What if memory isn't just, a static file locked away in our brains? What if it's something far more adaptable, maybe even fluid? And what if our whole understanding of identity, maybe even life and death, is actually ripe for a radical reevaluation based on, well, some pretty new science? Today, we're diving deep into a truly captivating paper. It's called A Biological Basis for Human Reincarnation by J. Alford, published just this year, 2025. Now, before you start picturing ancient mystics or anything, let's be clear, this isn't about folklore. This is actually about some groundbreaking recent experiments, specifically in bioelectricity, that might just offer a scientific lens on stuff many people have considered, well, inexplicable.
Right. Our mission today is basically to unpack how these pioneering discoveries, especially around bioelectrical fields, are forcing researchers to fundamentally rethink things. Things like, where do memories actually reside? And maybe even how life forms, or aspects of them, could persist across different physical bodies. We've pulled together some pretty diverse sources for you, and when you look at them through this lens, they paint a really astonishing picture. It's quite thought-provoking.
Okay, so let's start this journey into the weird world of memory. And our starting point isn't like a typical brain lab, it's actually with this tiny creature, the planaria flatworm. Michael Levin at Tufts University has done some incredible work here. What did these experiments show about where memory truly lives? Sounds kind of wild.
Oh, Levin's work with planaria is absolutely foundational for this whole idea. It's really key. So he taught these worms certain behaviors, right? Like how to navigate a maze, standard stuff. But then, and this is the kicker, he cut them into pieces. And here's the mind-blowing part. Each piece grew back into a whole new worm. And those new worms, they remember the behaviors their original parent worm learned.
Whoa, wait, each piece.
Each piece. It strongly suggests that memory isn't just stuck in a central brain. It's more like a dynamic electrical blueprint, something that can be copied, transferred, recreated. It fundamentally redefines where you, or at least your memories, might actually reside.
That's incredible. Okay, so if memory isn't just neurons firing in the brain,
Yeah.
what does Levin think is the actual, substrate for these memories? What's the mechanism? How does this seemingly impossible transfer happen?
Well, what's fascinating is how Levin's research points directly to bioelectrical fields. That's the proposed substrate. The mechanism involves the body's own complex electrical system. You've got tiny charged particles, protons, moving through special gates in cells' ion channels, creating these subtle voltage differences across cell membranes. Think of it like every single cell contributing to this huge living electrical grid. And crucially, it's not just brain cells, it's all cells. We're talking the 30 trillion cells in an average human body. They collectively generate and apparently store memories through these bioelectrical patterns.
Okay, hang on. How is it even possible to recreate a whole complex electrical pattern from just one tiny fragment? That seems counterintuitive.
Yeah, that's a really important question. According to Levin, when a planaria is cut, the overall bioelectrical pattern doesn't just disappear. It collapses and is downscaled, basically, to fit onto each individual piece. So every fragment, no matter how small, effectively contains the complete blueprint, the whole pattern, just smaller. Then, as that piece regrows, the pattern gets upscaled by all the new cells joining in, and boom, you get a complete organism again, complete with its original learned behaviors.
Wow. Okay, this seriously twists how you think about memory. But then it leads to an even more astonishing question, right? If memory can persist in pieces of an organism, what happens if the brain, the central command center, is completely destroyed? Can memory, maybe even the essence of identity, survive that kind of obliteration?
That's where the caterpillar to butterfly metamorphosis comes in. It's an incredible natural parallel. During that pupa stage, the caterpillar's brain essentially liquefies. It turns into biological goo, complete transformation. Yet the butterfly that emerges, it still carries memories and behaviors from its caterpillar stage. Like, they've shown they can remember avoiding electric shocks they experienced as larvae.
No way, even after becoming goo.
Even after the goo stage. It strongly supports this idea that memories are somehow imprinted everywhere within the organism's overall bioelectric field. The author of this paper proposes that while most of the caterpillar turns liquid, some highly organized cells remain solid. The caterpillar's bioelectric field, he suggests, downscales onto these remaining solid bits. And then it sort of morphs seamlessly into the butterfly's new bioelectric field, carrying those memories across the transformation.
Okay, this idea of memory living outside the brain, it naturally brings us to something similar, potentially happening in humans. You hear about these medically documented cases, right? Memory transfer in organ transplant patients. How does this biological fluidity, this sort of thing, show up in our own species?
Indeed, and the evidence here is pretty striking when you look into it. There are specific studies. A 2022 study in transplantology, for instance. They surveyed heart, liver, and kidney recipients. Get this. Nearly 90% of participants experienced changes in their personality, temperament, emotions, and food preferences after their surgery.
No,
90%.
90. That's huge.
Right. And then a 2024 study in Curious concludes something similar, that recipients might inherit not only new organs, but also the memories and personality traits of their donors.
Hmm.
So this isn't just like a weird anecdote here and there. It's empirical data suggesting our sense of self-likes, dislikes, fears might be encoded way beyond just our gray matter. It seems like it's throughout our physical form and potentially transferable.
Those studies are compelling. But are there any, like, really striking individual stories, maybe even outside of formal studies, that illustrate this idea?
Oh, absolutely. The anecdotes are often what really grab you. You hear stories like a child getting a heart transplant and suddenly developing this intense fear of water. Turns out the donor had drowned.
Mm.
Or there was a case of a health-conscious dancer, received a heart, and suddenly started craving Kentucky Fried Chicken like crazy. That specific craving was traced directly back to her donor's preferences.
Wow.
And probably the most famous anecdote is from Paul Pearsall's 1998 book. He's a neuropsychologist. He detailed the case of an 8-year-old girl who got the heart of a murdered 10-year-old. She started having these vivid nightmares, apparently seeing the murderer's face. And incredibly, her descriptions reportedly helped police identify and convict the actual killer.
That's incredible. Okay, so this has led to the cellular memory hypothesis, the idea that memories, personality traits, they're stored in organs like the heart, not just the brain. Now, if we connect this back to Levin's work on bioelectricity, the author of the paper suggests the bioelectric field is the real substrate for this cellular memory. So when an organ's removed, its field downscales onto it. Then, in the transplant, it fuses with the recipient's field, integrating the donor's memories. It really does sound like a kind of piecemeal reincarnation, doesn't it? Piece by piece.
Exactly. Piecemeal reincarnation is a good way to put it.
But then how small can this transplant be for memories to transfer? Could it be down to, like, just one single cell?
Well, it's a critical question, and it really pushes the boundaries, doesn't it? Think back to Thomas Hunt Morgan's discovery in the 19th century. He found 279 pieces of a planarian worm could regenerate, and each of those pieces was tiny, about 36 microns across. Now, compare that to a human heart cell, a cardiomyocyte. It's roughly 30 microns wide.
Wow, so basically the same size.
Pretty much. It strongly suggests, at least theoretically, that a single cell, or maybe a small clump of cells, could potentially hold a downscaled bioelectric field, a field containing retrievable patterns, maybe even memories. The possibility itself is just mind-bending.
Okay, this is leading us into even more astonishing territory. Let's talk about Ian Stevenson. He was a psychiatrist, a researcher at the University of Virginia, spent decades on this, studied hundreds of what he called reincarnation-type cases in humans. And what's really fascinating is his documentation of birthmarks, even physical features that seem to mirror injuries from claimed past lives.
Yeah, Stevenson's work is incredibly meticulous, very detailed, and yeah, highly compelling. He recorded cases like a boy with vivid memories of having his throat slit in a past life. And this boy had a very distinct reddish mark around his neck, exactly where that kind of injury would be. Another case, a boy claimed he died by gunshot suicide. He had 2 birthmarks, like scars, that lined up perfectly with where entry and exit wounds would be.
So how does the paper explain these physical marks?
The author hypothesizes that really high-impact events, things that cause significant physical distortion, like a bullet wound, they could potentially damage those tiny electrical gates of the cells, the ion channels, in a very specific pattern. This damage would modify the body's overall bioelectric field pattern. And then, if that field pattern, or a part of it, transfers, it could manifest as a physical feature in the recipient's fused bioelectric field. It's kind of analogous, the author suggests, to Levin's experiments where impacting a planarian could make it grow 2 heads. Changing the bioelectric blueprint creates a physical change. Seeing physical marks that seem to reflect past traumas is really quite remarkable.
Right. So according to this theory, when are these marks most likely to appear? Are there specific conditions?
Yeah, the author proposes the marks are usually clearest when 2 main conditions are met. First, a relatively short time lapse between the donor's death, or when the cell or organ leaves the body, and the recipient's birth or absorption of that material. Second, it seems clearer when the donor was relatively healthy and young at the time of their death. And interestingly, these conditions often match up pretty well with organ transplant scenarios too. and they seem to fit many of the cases Stevenson documented.
And there was another really powerful example Stevenson found, right, involving Burmese children.
Yes, that one's striking. He documented cases of children in Burma, now Myanmar, who had these incredibly detailed, uncanny memories. They remembered being British or American pilots from World War II, specifically pilots who were shot down over that region. And what was remarkable is that these children often had physical traits that were unusual for their families. Things like fair hair, lighter complexions, sometimes even distinctive facial features or foot deformities that seemed to match descriptions of the pilots they claimed to have been. This specific type of case really highlights the potential implications.
Yeah.
It's strong evidence, potentially, for the fusion of not just memories and behaviors, but also morphogenetic bioelectric fields. That's the part of the field that guides physical development, shapes the body. So you're seeing the physical structure, behavioral patterns, and memories apparently transferring from the pilot to the child. But, as the author rightly asks, it raises that crucial question. How did the Caucasian pilot's bioelectric pattern get into the Burmese child's body? How did the child become a human chimera? And that's where this whole concept of chimerism becomes absolutely central.
Okay, yeah, let's unpack that. Human chimeras, it sounds like something out of mythology, but it seems key here. What exactly is a biological chimera?
Right, it sounds mythical, but it's a real biological phenomenon. Yeah. Biological chimeras are simply life forms that have body parts with different genetic origins from two different individuals. And it's documented in humans, not just theory. The first reported case was back in 1953. Then there was a famous case in 1960, a baby girl born in Seattle. She had one brown eye, one hazel eye. Internally, she seemed to have an ovary on one side, a testicle on the other, and she had two different blood types. Genetic tests confirmed her cells came from two distinct individuals fused together.
That's incredible.
And it still happens. More recently, 2015, a man in Washington state took a paternity test. It showed he was genetically his son's uncle, not his father. Turns out he had different DNA in his saliva compared to his sperm. A clear case of human chimerism.
Okay, wow. So how do these chimeras actually form? What are the different ways this can happen, naturally or artificially?
There are a few known ways. You've got twin chimerism. That happens in the womb when one embryo absorbs some cells from a fraternal twin that didn't survive. Then there's fusion chimerism, where two separate fertilized eggs, two zygotes, fuse very early on and develop as a single embryo. But crucially for this discussion, there's also artificial chimerism. This happens when someone receives cells from an external source, blood transfusions, bone marrow transplants, and organ transplants. Organ transplants essentially create fusion chimeras. You're directly merging tissues, cells, and potentially, as we've discussed, the associated bioelectric information and memories. So it definitely shows that cells carrying all this potential information can be delivered from outside sources into a new body.
Which brings us to maybe the most mind-blowing example, taking this from multicellular life down to what seems like almost unicellular, the dog cancer example.
Oh, the canine transmissible venereal tumor, CTVT, is one of the most bizarre things in biology. It's a specific cancer found on dog genitals, and it literally transfers between dogs during mating, like an infection. But here's the thing. The tumor cells are genetically distinct from the host dog they're growing on. They are effectively cells from another dog.
Wait, from another dog?
Yes. Scientists traced its origin back to the cells of a single dog that lived around 11,000 years ago, near the end of the last ice age. So in a very real sense, this one original dog has been partially reincarnating or persisting through its cancer cells for millennia. The source material notes this cancer is now basically living as a unicellular, asexually reproducing pathogen. It's a fascinating perspective. Cancer cells sometimes viewed as cells rejecting the cooperative multicellular society and reverting to a more solitary unicellular lifestyle, but still carrying the genetic legacy.
That's a profound idea. Does this concept of toggling between unicellular and multicellular forms, does that have implications for how we understand biological death itself?
Yes, absolutely. And this ties back into Michael Levin's more recent work with xenobots. These aren't robots in the mechanical sense. He took skin cells from frog embryos and grew them in a dish. They self-assembled into these tiny, autonomous sort of balls of skin that could swim around, react, even repair themselves if damaged, multicellular blobs. But then he found if you broke these multicellular xenobots apart into individual cells, those cells could later reaggregate and reform into new, multicellular xenobots.
They could reform.
Yes. Levin himself says the original organism is no more. The individual cells live on and eventually get back together into a multicellular form that swims and reacts to stimuli. It really blurs the line, doesn't it, between what we think of as death of the organism and just a change in form, with the individual cells persisting.
So what does this imply for the very concept of death? Is it not always the end?
Well, Levin hypothesizes that maybe life forms can, in principle, toggle between unicellular and multicellular states. It could be part of the life cycle. He explicitly states, there is nothing inherently impossible about such a life history. And he suggests there could even be an evolutionary advantage. Imagine cells from a body that's dying. They could continue their journey through the world as unicellular organisms when their body is no longer viable as a whole. He even talks about them potentially rebooting their multicellularity by aggregation later on. The individual cells and the information they carry might persist beyond the death of the larger organism.
That's radical. How could this possibly apply to human death? We usually see that as pretty final.
Well, think about it. The death of a complex human body isn't like flipping a switch. It's a process. Different cells die at different rates over hours, even days. So the author of this paper posits, what if some parts, maybe a collection of cells from the heart, for instance, haven't fully died yet when the body is pronounced dead? Could the body's bioelectric field downscale onto those surviving cells? If so, the body plan, the memories encoded in that field, might still be retrievable, at least for a time. And it's fascinating, the human heart isn't just a pump. It has around 40,000 neurons of its own. It forms an intrinsic cardiac nervous system that can actually process information and modulate memory.
40,000 neurons in the heart.
Yep. And individual heart muscle cells, cardiomyocytes, are incredibly resilient. They can survive and even continue beating independently for weeks after being separated from the body, like in a petri dish.
This idea of persistent life units or memory-carrying particles, it actually echoes some much older, maybe more esoteric ideas, doesn't it?
It really does. You find fascinating parallels over a century ago. People like Helena Blavatsky wrote about life atoms. She believed these were released during human death and contained memories and behavioral tendencies. She even suggested ancient Egyptian mummification was an attempt to prevent these atoms from transmigrating and carrying identity elsewhere, while cremation, she thought, helped disperse them and prevent identity drift.
Interesting.
And Annie Besant, around the same time, called them permanent atoms. She believed they preserve powers of vibrations, which sounds a lot like frequencies or waveforms carrying information, like memories. And significantly, she believed these atoms resided around the heart, noting that the heart region is the last part of it to break up. Besant also talked about injuries to the physical body, creating lesions in a lower energy invisible body, which sounds strikingly similar to the idea of damage altering a bioelectric field pattern. It's remarkable how these older concepts seem to prefigure some of these modern scientific ideas.
And then, totally different field, but what about things like orbs? You see them in paranormal photos and videos all the time. Any connection there?
Well, it's speculative, of course, but the paper does mention orbs. These are those light anomalies, usually spherical balls of light that appear in recordings. They often move rapidly, and some descriptions compare their movement to amoebic life forms in a petri dish, which, interestingly, sounds a bit like descriptions of Levin's xenobots swimming around. So the paper raises the question, it's just a question, could some of these orbs potentially be, say, vibrating living human cells existing in a temporary unicellular or near unicellular state containing a downscaled bioelectric field with a coded body pattern and memories? And could they, theoretically, be captured, transplanted, and fused with another body, creating a human chimera? It's definitely out there, a provocative thought linking these different phenomena.
Okay, so let's pull these threads together. Based on all this, the planaria, the transplants, the chimeras, the toggling life forms, what does the author actually propose as the mechanism for how this kind of reincarnation might happen naturally, outside of a controlled setting like a transplant?
The core proposal is this. If one cell, or maybe a small collection of viable cells, escapes from a dying body before cellular death is complete, they could potentially survive independently for some period, maybe as a kind of unicellular or near unicellular life form carrying that downscaled bioelectric field. Then, if these cells encounter another living human, and if they happen to resonate, maybe through biological compatibility, similar frequencies, who knows? They could potentially be absorbed by that individual and integrate, continuing their existence now as part of a new multicellular life form.
Absorbed. How would cells actually transfer from one body to another without, surgery? That seems like the biggest hurdle.
Right, that's the key practical question. The paper theorizes about specific scenarios.
Hmm.
Think about those pilots shot down midair or people suffering massive trauma, especially to the chest area. It's conceivable that human cells, particularly resilient ones like heart cells, could be aerosolized or disseminated into the environment, sprayed out basically. Now imagine if some of these airborne cells, carrying their bioelectric field and memories, land on an open wound of another living human, maybe a child playing who often has scrapes and cuts. The cell could potentially enter the bloodstream, travel through the body, and perhaps get deposited in the recipient's heart. If it's biologically compatible enough, maybe it could fuse with the recipient's own cells rather than just being destroyed by the immune system.
Huh. So like a microscopic transplant delivered by chance.
Essentially, yes. The paper calls this specific scenario low-energy horizontal reincarnation. It's horizontal because both the donor and recipient bodies are, on the same physical plane, same energy level,
Yeah.
no spiritual dimensions required. And it's low energy, presumably because it doesn't involve massive energetic shifts. It's framed broadly as a kind of micro heart cell transplant, leading to microchimerism. And this is proposed as the potential biological basis for those reincarnation cases Stevenson studied, the ones with specific memories and birthmarks. It offers a possible natural mechanism beyond twin or fusion chimerism in the womb for how these transfers could occur, with organ transplants being the artificial version.
So it provides a way, at least theoretically, for memories from someone geographically distant, genetically unrelated, to end up in another person's body.
Exactly. It bridges that gap. It suggests a biological pathway, however improbable any single instance might be. So if you look at the bigger picture here, this whole line of research, this deep dive, it really suggests something profound, that memories, maybe even aspects of our identity, might not be solely locked into one brain, one body. They might exist within this incredibly adaptable bioelectric field, a field that can potentially downscale, transfer, survive transformation, and even integrate into new forms.
Wow. So what does this all mean for your own understanding of memory, identity, and, well, the very concepts of life and death? It really makes you reconsider that intricate dance between our electrical selves, and consciousness, doesn't it? Definitely something to mull over next time you hear one of those weird coincidences or maybe experience a sudden, inexplicable craving that feels oddly not quite your own.